He will join in a few moments.
Okay. Then should we start now?
Yes, sir. Yes, sir.
So, a very good morning to all the participants and distinguished speakers of this session. This is the day 2 workshop number 3 which is based on genomic-assisted strategies for climate resilience crop. It is being organized by AKS University Satna, and today we are privileged to have 3 eminent speakers from our country, and they are Dr. Anil Kumar Singh from IIAB Ranchi, Dr. Rajiv Nayan Bahuguna from Rajendra Prasad Central University, and Dr. Srinivasan from Chennai. Our first speaker of the day is Dr. Anil Kumar Singh. I welcome you, sir, in this workshop. And before we request him to start his presentation, I would like to introduce Dr. Anil Kumar Singh. The first introduction that I would like to give for him, that I have a very long association with Dr. Singh, almost 23 years. It does not look like that, but yesterday I was counting that how many years has been for. So 23 years he is associated to me, and it was a culture in our university that we have learned many things from our senior, and I am very privileged to have a senior like him, and he has guided us many things like a teacher. So I welcome you, sir. Dr. Anil Kumar Singh is presently working as senior scientist and head School of Genetic Engineering at ICR, Indian Institute of Agriculture Biotechnology, Ranchi. He obtained his MSc in 1999 and PhD in 2004 with major in plant physiology from GB Pant University of Agriculture, Technology and Sciences, Pantnagar. And thereafter, he joined International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi, as a postdoctoral fellow. Dr. Singh joined CSIR Institute of Himalayan Bioresource Technology, Palampur, as scientist in 2011 and continued till December 2015. Dr. Singh has made outstanding contribution in the area of plant functional genomics, and he has characterized transcriptome of several plants including Capgana jubata, horsegram, saffron, apple, tea, mustard, stevia, and wild chickpea, and identified several important genes from these plants. He is a pioneer on understanding the effect of climate change on apple bud dormancy, and for this contribution, he has a global recognition. Dr. Singh has been instrumental in genome sequencing of several commercially important bacterial species identified from cold desert area of Himachal Pradesh. Dr. Singh has published more than 60 articles in peer-reviewed journals with high impact factor, and his cumulative impact factor is more than 125, which is a very significant impact factor, and his citations are more than 1400 plus. He has already filed one international patent. Dr. Singh is now serving as an editor of +1 Plant Physiology Reports. He is also guest editor of Tree Physiology, and he is selected as a member of NASI, that is National Academy of Sciences India, and also he is a member of Plant Tissue Culture Association of India, that is PTCAI. So with this very highly weighted and brief introduction of Dr. Singh, I welcome him again, and I request you, sir, to please start your presentation.
Thank you, Dr. Pradeep, for very nice introduction, and I would like to thank organizers for giving me opportunity to present some of my work. I'll just share my PPT.
Dr. Arpit, Engineer Arpit, can you guide Dr. Singh for sharing the screen?
Yes, sir.
Please.
Sure, sir.
Yeah.
Dr. Anil, sir, just
yes.
on the right screen of the blue jeans meet, on the right side, the fourth icon is there.
Sharing panel.
Yes,
sharing panel.
Yes.
Yeah.
Just click on that. Now it will
when I
ask to
saved it,
download the extension. Fourth
okay,
icon, sir.
yeah, I've done it. So
Okay.
I need
Now
to
it
download
will
this
ask.
extension.
Uh, yes. Hardly 30
Okay,
seconds.
yeah.
So once it is present on the Google, just again you go for the sharing option, and you
Okay.
will see the sharing screen, and just click on that. You will ready with your presentation.
Yes. Now I can see.
Yeah, it's visible now, sir.
Okay,
Yes, sir. It's visible.
okay, fine.
Thank you,
Uh,
sir.
so the title of my talk today is Integrated Omics Approaches for Understanding Plant Response Under Climate Change. And the title of your workshop is also in the same line. So I thought this is most relevant for this workshop. So for benefit of students, so you have been hearing about climate change, but what is climate change? As you can see, the temperature has been rising since a very long time, more than 150 years. But the increase in temperature was, not that rapid as compared to last 20, 50 years. If you look at this yellow line, so problem started somewhere here. People have started realizing that the temperature is rising very steeply. So then people have initially termed it as a global warming, but later they realized that it is not only global warming, but whole climate is changing. Uh, so and due to this climate change, overall climate change, which includes global mean temperature rise and also increase in carbon dioxide and some other gases, the global mean temperature has increased around zero point 0.7 degrees centigrade during last 50, 60 years, and it may the alarming situation is that it may increase by 3.7 to 4.8 degrees centigrade by the end of this century, and which is very significant. And due to this climate change, precipitation will increase by 4 to 5% by 2030 and by 10 to 15% by 2080. But the problem is that intensity of this precipitation will increase, and the distribution is suspected to be uncertain, as we have already started realizing it. Uh, and due to this, sea level will rise by 20 centimeter. This is a huge number. 20 centimeter means most of the coastal cities globally, not only in India, but globally, most of the coastal cities will be submerged. And this will lead to retracting of glaciers and change in overall seasons. And this change in season will impact, whole agriculture system. Uh, And due to climate change, the intensity of weather extremes is, suspected to be changed. There will be more cyclones, heavy rains, heat waves, flood, drought. And the problem is that in one cropping season, there may be more than one extreme events. There may be drought during initial phase and then submergence or flood at later stage. And there may be heat stress also in the same cropping season. So we need to develop plants which can sustain uh, high temperature, drought, and flooding in a same season. So plants need to be developed with multiple stress tolerance. So in our laboratory, we have been using these integrated omics there have been a lot of whole genome sequences available in public database. So one of the approaches is to mine uh, that genome sequence and mine gene families, and then you can do analysis of these families and do expression profiling, and then you can identify a handful of genes which may be used for developing climate resilient crops. And the gene resource which we have developed through these approaches have been deployed in some model plant system like Arabidopsis and tobacco, and also in some crop plants like potato and rice. So this is in a nutshell what my lab has been doing for last 15 years or so. But today I'll be focusing mainly on two examples from my lab. Now, first one is understanding molecular regulatory mechanism of chilin-mediated bud dormancy release in apple, and second one is identification and characterization of important genes of nitrogen use efficiency in mustard. So first, the apple story. As we all know that apple is a very important fruit crop, and mainly this crop is of temperate regions. But in India, geographical location and environmental factors of mid and high altitude regions of Uttaranchal, Himachal Pradesh, and Jammu Kashmir make it suitable for apple cultivation. And in apple, during winters, seasonal dormancy is an adaptive mechanism to avoid damage due to chill in temperature. As we know, many crop plants cannot survive chill in temperature, but for apple, this chilling is essential for development of normal fruit. So this chilling period is essential for normal phenological traits in apple. And this stream I'll explain little more in little more detail, because this is important to understand before going into the detail of my study. During winters, plants accumulate chilling temperature, and 1 hour of temperature equal to or less than 7 degrees centigrade is considered as one chilling unit. So if during winters plant accumulates sufficient chilling units, that is more than 1200, then at the onset of spring, the dormancy breaks and the quality and yield of apple is good if that sufficient chilling units are accumulated. But if the chilling units are not sufficient, then after dormancy break, the quality and yield of apple fruit is not that good. So this indicated that chilling during dormancy plays important role in determining apple fruit quality and yield. When I started this work, there were a lot of news articles in newspapers and in media that in mid altitude regions like Kullu in Himachal Pradesh, people are now realizing that apple cultivation is not profitable. So people are shifting to off-season vegetable cultivation. So this made me think that what is the reason why these plants are not able to develop good yield and fruit quality due to rise in temperature. Uh, so we designed our study and we selected two locations, one at Kullu and another above Manali. This Palchan, Manali was the second location, and we selected temperature data of mean maximum and mean minimum temperature data from the stations located in Kullu and Manali, and we found that there was significant difference in mean minimum and maximum temperature during winters. And we also calculated chilling units, and we found that the chilling units in Kullu are not sufficient, while in Palchan, Manali, the chilling units are sufficient. So we compared these two locations. My students used to visit Kullu and Manali at regular intervals to collect samples in liquid nitrogen. So this was a huge task. So they collected 8 samples of dormant bud, apical and meristematic both, apical and vegetative bud both, and 6 stages of flower and 6 stages of fruit development. So total 20 samples from each location were collected, and we analyzed these samples through transcriptome analysis, and we developed more than 90 million reads and did all sorts of routine analysis to identify up and down-regulated genes. But I'm not going into the detail of up and down-regulated genes. Uh, when we analyzed our data closely, we found that some Madsbox transcription factors, which are dormancy associated, which are known as DAM, and FLC-like genes were found to be downregulated along the dormancy release. So this is a normal expression pattern of these genes. It is known that as chilling is accumulated, these DAM genes, expression of DAM genes slows down. When we compared expression in high chill, this DBH, STH, this is for high chill location and this is for low chill location. So you can see the reduction in expression is steep in high chill location, while it was not that steep in low chill location. Similarly, the FLC-like genes expression is steep down in high chill, while it was zigzag in low chill condition. And for this FT, floral locus T gene, flowering locus T gene, the expression is supposed to go up when the dormancy is broken. So you can see there is a steep rise in expression of this gene during dream tip, means at dormancy break, while it was not that significant in low chill location. So we found that there is a significant difference in expression pattern of DAM or FLC and FT genes, which are members of Madsbox transcription factor family. So we thought to identify whole family of Madsbox transcription factor genes in apple, and then we compared phylogenetically Madsbox transcription factors of apple with other diploids, and we divided into different classes. And when we closely analyzed this dendrogram, we found that apple contains 4 FLC genes and 6 DAM genes. And to our surprise, these FLC and DAM genes were not even reported in Apple genome sequencing paper, which was published in Nature in 2010 or 11. So this was the first report that Apple contains FLC-like genes and DAM genes. So before this, there was a debate whether Apple has these genes or not. Then we plotted expression of all the transcripts belonging to Madsbox transcription factor family in our transcriptome data, and we could found that some of the genes have a specific expression in flower and fruit, but not in this green color means low expression and red color means high expression. So we found that some genes have high expression in flower and fruit, while lower in birds. And so on. And based on this, we plotted a model of all the Madsbox transcription factor genes, that in which condition, which stage, which gene goes up and which gene goes down. And this we published in scientific report. So this was the first report basically from Apple that what is the expression of whole Madsbox transcription factor family in various stages of apical bud dormancy, flowering, and fruit development. So to extend this transcriptome data analysis, we did weighted gene flow expression networks analysis, because in transcriptome analysis, you obtain thousands of genes upregulated and thousands of genes that in downregulated. So sometimes people are confused, how to interpret this data. So there is a approach called WGCNA, by which you can identify a handful of genes which are putative master regulators of the process. So we did this analysis and we identified different modules in low chill and high chill conditions. And based on statistical analysis, we narrowed down to two modules, yellow and purple module, which are significant in our data. And when we plotted expression of those genes involved in these modules, this is yellow module and this is purple module. So you can see there is a significant difference in expression pattern of genes in dormant bud and green tip. Green tip means where expression, where dormancy is just broken, and dormant bud means when it is in dormancy. And similarly, in high chill condition also, there was a significant difference in these stages. So this color coding shows you how the expression is changing in different stages. And when we did cluster analysis of all the genes involved in these modules, we identified some hub genes and some genes with intramodular connectivity. Hub genes means the gene which is sitting at the core of this network, or which is connected to most of the genes in the network. And when we did enrichment analysis, there were very large enrichment plots were obtained, but just to show the pattern which is happening in this cluster, there were some post-embryonic development terms and some other important geoterms were highly enriched in yellow cluster, while it was not in that much significant in purple cluster, but post-embryonic development geoterm was enriched in this also. So then we identified the genes, 5 hub genes and 5 intramodular, high intramodular connectivity genes, and we found that many of the genes are either transcription factors or the genes which are known to be involved in dormancy rate, flowering, and fruit development. And similarly, in purple cluster, we identified some important genes. So the gist of this analysis is that we could identify a handful of genes which can be functionally characterized to understand their role in chitin-mediated dormancy release in apple. When we analyzed our data, we found that there are several genes of DNA methyltransferases and DNA glycosylases which are involved in DNA methylation and demethylation. They were differentially expressed. So just to see, so this suggested us that epigenetic regulation also plays important role in chitin-mediated dormancy release. So just to see what is happening in terms of DNA methylation, we did MSAP analysis, which is based on methylation sensitive amplification polymorphism is dependent on these isosizomers MSP1 and HPA2 with different sensitivity to cytosine methylation. So we performed this MSAP analysis. This is just a representative gel and bands which are marked. So my student did a lot of gels, and after exploring, we found that total methylation in high chill condition is reduced as the chilling is accumulated from dormant bud to fruit set stages. The total methylation is reduced in high chill, but in low chill condition, there was no change in total methylation. So as you know that during dormancy, metabolic activities goes down. So we speculated that the mode of reducing the metabolic activities is through DNA methylation. And as the chilling is accumulated, the genes are slowly released from methylation and induced. And the flowering and fruit set starts. While these genes were not released by from methylation in low chill condition, and suddenly when dormancy is broken, so that affected fruit set and fruit quality. So this whole exploring analysis suggested that apple bud dormancy is maintained through DNA methylation, and chilling association is associated with gradual demethylation of DNA. And when we sequenced all the bands, we found that these genes are involved in various metabolic activities which are differentially methylated. And then we plotted expression of those genes. We found that demethylated genes are generally highly expressed. This red color means high expression, and demethylated genes are, you know, very low. They have very low expression, while only few have high expression. So, to conclude this apple story, the transcriptome analysis during dormancy release and fruit set in apple under differential chilling identified some hub genes like early flowering 7, RAF10, ZEP4, and APOX, which may be involved in regulating chilling-mediated dormancy release and fruit set. And Madsworth's transcription factor family was identified, which contains 142 proteins. And through this analysis, we identified 6 putative DAM genes and 4 putative FLC genes. And changes in methylation pattern during dormancy release and fruit set under differential chilling availability was identified, which suggested that adequate chilling availability during dormancy period is associated with demethylation of apple genome along the dormancy release and fruit set. So this was all about Apple story, and this Apple work has been recognized globally. After we published this MSAP paper, there were many other reports citing our paper that similar mechanism exist in other plants like sweet cherry and almonds and some other plants also. So now second story is about identification and characterization of 3 regulators of nitrogen use efficiency in mustard. So as we know that during Green Revolution was brought about mainly by high yielding varieties and high applications of chemical fertilizers. And after Green Revolution in India alone, the consumption, production, and import of nitrogenous fertilizers has increased several fold, and it is predicted that half of the synthetic nitrogen fertilizer ever used has been utilized since 1985. So, on one hand, this has increased productivity, but on other hand, there are several downsides of excess nitrogen application in agriculture fields. It causes nitrate leaching. The excess nitrate which is applied to the field is leached to groundwater, and it contaminates water bodies also, causing eutrophication. And the nitrous oxide which is released cause damage to ozone layer, and it cause ammonia volatilization and acid rain. So on one hand, nitrogen fertilizer has a beneficial effect on plant productivity, but on other hand, if it is used in excess, it has several downsides. So this is alarming because global warming potential of nitrous oxide is about 300 times more than carbon dioxide. So reducing excess nitrogen fertilizer use is very important. So that's why we need to develop nitrogen use efficient crops. And why we have chosen brassica, because we know that brassica is a very important oilseed crop for India, and it contributes nearly 30% of total oilseed production. And the main thing is that requirement of nitrogen per unit of yield is higher than most other grain crops. So brassica needs very high amount of nitrogen fertilizer. So as you can see, edible oils constitute a major portion of agricultural commodities which are imported. So we are still not self-reliant. Now we are talking about Atmanirbhar Bharat. So we are not Atmanirbhar in terms of edible oils. We import a lot of edible oil every year. So that's why we need to understand the mechanism of nitrogen use efficiency, and we need to develop high nitrogen use efficient crops, which will reduce input cost. It will create less environmental pollution, less nitrogen loss, and efficient use of nitrogen fertilizer for plant growth and development. So when we started this work in 2011, there was a report by Ahmad et al. 2008, where they have screened some mustard varieties in field, and they found that Pusa Jatisan has lowest nitrogen use efficiency, while Pusa Bold has highest nitrogen use efficiency. So we chose these two varieties of Brasitha to compare their response in terms of various nitrate treatments. So we chose 4 nitrate treatments total: 0, 0.25, 2 millimole, and 4 millimole potassium nitrate. And at various time intervals, we have collected samples and did transcriptome analysis of total 42 samples. This 0.25 millimole potassium nitrate was chosen because nitrate also acts as a signaling molecule. So to understand its effect as a signaling molecule, very low amount for very short time was given, which was 0.25 millimole for 20 minutes. So in addition, we have we have analyzed several growth parameters, and we found that Pusa Jatisan performed better than Pusa Bold performed better than Pusa Jatisan at low nitrate concentration. And we also analyzed nitrate reductase activity. And NR is the main enzyme of nitrate assimilation. So we analyzed nitrate reductase activity and total nitrate content also in both the varieties. And we found that Pusa Bold has higher nitrate reductase activity than Pusa Jatisan in most of the treatments. So as I told you that we have done transcriptome analysis of 42 samples. So I'm not going into the detail of all the routine analysis of transcriptome, just to show the screenshot what is happening in these plants. So these are enrichment plots. At low nitrate, you can see there are many geoterms which are highly enriched in Pusa Bold, like phenylpropanide metabolic process, cell wall organization, response to chemical, response to hormone stimulus, while these terms were very lowly enriched, or you can say not enriched in Pusa Jatisan. Similarly, at 2 millimole nitrate and 4 millimole potassium nitrate, very important geoterms were enriched in Pusa Bold, but this was not the case in Pusa Jatisan. So we found that there is a significant difference in molecular level screenshot of these two varieties under low nitrogen treatment. Then we identified whole genes of nitrate uptake, nitrate assimilation. Sorry for this slide. There is something that is not visible, but this is basically nitrate uptake and nitrate assimilation. And as we know that carbon and nitrogen metabolism also interact, so we have also identified genes of carbon metabolism, and we found that in Pusa Bold, several genes are induced at very early time points. That this is 0.25 millimole nitrate for 20 minutes, and this is 2 millimole nitrate for 2 hours. So we found that many genes are induced at very early time point, while in Pusa Jatisan, this was not the case. They were not induced at early time point. And in later time points also, there was a significant difference in expression pattern of these genes. And then we did WGCNA analysis for this data also, as we did in case of Apple. And we identified that 4 modules were statistically significant in Pusa Bold, and only 1 module was statistically significant in Pusa Jatisan in response to nitrate. And then we did cluster analysis and identified hub genes. And when we plotted expression of these hub genes in various treatments, we found that in Pusa bold, most of the hub genes have high expression. Uh, this red color means high expression. So you can see most of the hub genes have high expression. But in Pusa Jatisan, there were only two genes which are induced, while rest three genes uh, were not induced. So this gave us, indication that what is happening in these varieties, why Pusa Jatisan is having low nitrogen use efficiency and why Pusa Bold is having high nitrogen use efficiency. So then to further analyze this data and further understand the effect of various stresses, we cloned all 26 genes of nitrate uptake and assimilation pathway. Sorry, there is some disturbance in naming of these genes, but this plot basically gives you indication about induction of those genes in various other abiotic stresses like salt, osmotic, cold, and heat. As we know that all these abiotic stresses affect plant productivity, so we found that the expression of several important genes, which are marked here, uh, are having very low expression in these abiotic stresses uh, at 1 hour and 24-hour both. So this paper in PLOS One we published with the title that abiotic stresses downregulates three genes of nitrate uptake and assimilation pathway, and this paper is highly cited because there was no study in any plant before this that what is the effect of abiotic stresses on nitrate uptake and assimilation pathway. So to conclude this analysis, transcriptomics study helped us to understand molecular network that mustard plant use to integrate metabolic, cellular, and developmental processes to adapt to change in nitrate availability. And flow expression network analysis identified several putative master regulators, and we have cloned 26 genes spanning whole pathway of nitrate uptake and assimilation. And pyrimidine of NRT, this is a nitrate transporter, NR, nitrate reductase, and GS, glutamine synthetase. So these genes are very important for nitrate uptake and assimilation. So we thought that pyrimidine of these genes may increase nitrogen use efficiency of brassica. So we have submitted a proposal in SERB DST for this. So we will be developing transgenic plants over expressing NRT, NR, and GS to increase nitrogen use efficiency. And functional characterization of hub genes, hub transcription factor genes may provide insight into the regulatory mechanism of nitrogen use efficiency in brassica. So this work is also being done in my lab. So to acknowledge, I thank the director of my present institute and my past institute, IHBT Palampur, and I acknowledge funding from CSIR, DBT, SERB, and DST, and my group. Gulshan has done most of the Apple work, and Parul has done this brassica work, and Khushbu has helped in bioinformatics. Usha, Preeti, and Shivalika, they also helped in bioinformatics and some wet lab experiments. So thank you so much for your kind attention, and I welcome some questions.
Thank you very much, sir, for a very nice presentation in both the fields of nitrogen use efficiency and chilling stress. And your presentation gives and shows your significant contribution in both the fields. I congratulate you for that significant research in that field. We are waiting for some questions. And till then, I have one silly question, sir.
Yeah.
Actually, we are working very hard. The main scientific committee is working very hard and developing novel genes. Discovering novel genes and developing new plants against the climatic change.
Yeah.
Question is, suppose we have gone through those gene interventions, and suppose if climate change is reversed,
Sorry.
if the climate change is reversed in the coming time,
Okay.
like this year we are experiencing a little like that. So will those plants perform same, or they will perform in a different way?
No, I think in some places plants perform normal when situation is normal, but if you develop transgenic plants with stress inducible promoter, so the gene will express only when there is a stress. But
Okay.
if you are integrating various approaches, various pathways you are accumulating in one plant, so the response may be different.
All right.
And if you are using constitutive promoter, these genes will be constitutively expressed.
Okay.
But there are some concern in some plants. There are reports that constitutive expression has some yield penalty, or some plants waste a lot of energy in expressing those genes when they are not required. So we need to have very regulated expression of those genes. They are expressed only when there is a need.
Their physiology may be stable, or it may be differ according to some gene behavior.
Yeah, definitely it will differ.
Okay.
Not whole physiology, but the process which we have targeted.
Okay. That part. So we have also with us the organizing secretary, Dr. Kamlesh Chore. I welcome you, sir. Dr. Chore, I welcome you.
Yes, sir. Thank you. Good morning, sir. Actually,
Okay.
I am in this room very late, but
Okay.
I welcome all the guest speakers for today's session. I was just listening the lecture of Dr. Anil Singh. It is very path-breaking lecture, and
Yeah.
we are learning so many things by him. Sir, thank you very much for accepting our request to join us as a speaker of eminence. And
Thank you so much. My pleasure.
thank you, sir. And you have given time on Sunday. So thank you.
Yeah, that's a challenge sometimes.
That's
because
true.
because because with
Yeah, my daughter is continuously knocking the door.
yeah, right,
Yeah.
sir. So thank you. Thanks a lot, sir. Sir,
Yeah.
over to Pradeep.
Thank you.
Yeah.
So there are no questions from the audience, I think. So, yes, sir. You want to say something, Dr. Chore?
I have one question from Anil
Please,
sir.
you can.
That if we are inoculating certain microorganisms in the root, so
Yeah.
that we can alleviate this kind of abiotic stresses sometimes in the plant with the help of certain microorganisms.
Yes, there are some reports. For drought particularly, there are some reports that some WAM fungus, they help in absorbing moisture from subsoil and bringing it to the near to the root. And there are some other reports also. And recently we have also started working in the similar area. We are trying to identify some important microbes of chickpea and lentils, which are tolerant to acidic soil. Because in this eastern and northeastern area, a lot of soils are highly acidic. So we are now working on to identify some beneficial microbes which are acidity tolerant.
Okay, sir.
Yeah.
Very, very good, sir. Thank you, sir. Thank you very much.
Thank you.
Yes,
Okay, if
Pradeep,
there are no further question, Dr. Chore, you want to say something?
No, sir. Now you convene your session here.
Okay. So, if there are no further questions, then I must thank Dr. Singh. Sir, we are very grateful that you are here, and we are very privileged to have a speaker like you. And we have gone through your significant contributions in the field. Thank you very much, and I hope that in future you are will continue to help us in the same way.
Sure.
Thank you very much, sir.
Yeah, thank you.
Thank you. So, in the continuation, we have next speaker, Dr. Rajiv Nayan Bahuguna.
Yes, sir.
Uh,
Good morning, sir.
Dr. Rajiv, are you here?
Yes, sir. Can you hear me?
Yeah,
Am I
I can hear
audible?
you. You are perfectly audible and visible now. So, Dr. Rajiv is a very nice person, and then he is a very excellent scientist. He is presently working as assistant professor in Center for Advanced Studies on Climate Change in a center of Central University, Dr. Rajendra Prasad Central University of University Pusa, Samastipur, Bihar. He has a great academic profile, and he is a very significant contribution in the field of genomics throughout his career. He is a DST Young Scientist in the Department of Plant Physiology, IRA, New Delhi. He was a research scientist in School of Life Science, JNU, New Delhi. He worked as a postdoctoral fellow in IRI, International Rice Research Institute, Manila, Philippines. And he again worked as a postdoctoral fellow in Department of Plant Physiology, IRI, New Delhi. He has many awards to his credits like RDS Anna Gold Medal Award, ISPP Young Scientist Award, and DST Fellowship to his credit. He has given many presentations and many publications to his credit. His impact factor, cumulative impact factor, is more than is having a very high impact factor, and he is having a peer-reviewed journals publications almost 24. And at this age, he has gained so much experience, and today he will enlighten us with his lecture and talks. So I welcome you, Dr. Rajiv. You can continue with your presentation. Thank you.
Pradeep, before Rajiv starts, I want to compliment him for very nice achievements, and I look forward to listen to him after a very long time, after Pantnagar, when he was MSc student.
Sure, sir.
About 18-20 years.
Yeah. No, not more than that, sir. More than that. Rajiv, you आप जानते हैं ना अनिल sir को? I hope you know him.
Yes, sir.
Yeah.
He patient for us, all of us, actually.
Welcome, Rajiv.
So can
Rajiv, you can start your presentation. Huh?
Can you see my screen, sir?
Yeah, perfectly. It's perfect.
Okay. Okay, so thank you so much. I would like to thank first of all the organizers of this event, and this is very nice opportunity for me to present my work. As I go through all the by-eminent speakers previously, the most emphasis was on the genomics area. Genes were being used to achieve that's how we are addressing resilience and cross of climate change. In this presentation, I would, however, focus on another aspect of this kind of area where I would be focusing more on the phenotyping of crops for stress tolerance. So that was the major area of my postdoc, and currently here at this place. So this is the content. Basically, we will discuss about climate change and crop production, and then we will discuss how phenotyping is connected to genomics. And we talk about the stress and mostly about the stress combinations of this. Dr. Anil has discussed during his presentation that climate change plants will, particularly crops, will be facing most of the stresses. Means more than one or two stresses in a single crop cycle. So it would be mostly about the stress combinations and not just the isolated stress. Then we would be discussing phenotyping of crops for stress combinations and phenotyping of means role of phenotyping basically in the area of genomics. Two approaches we have reviewed actually discussing those two approaches mentioned as forward and reverse approaches, and then a few take-home messages, and I will then conclude my presentation. So climate change and am I audible, sir? Hello,
Audible, Dr. Rajiv?
am I audible?
You are audible and visible both.
Okay. So I don't know from where it was missed actually. So shall I start from the first slide or what?
Yeah, you can start from the beginning because from the second slide you were left.
Okay.
Okay.
I hope it's coming now.
Yeah. It's very visible.
Okay. Okay, so this is about the content we would be discussing a few points which are mentioned here, like climate change and its impact on global food production. Then I would be connecting how phenotyping is now considered as phenomics, and discussing about stress, which we were studying in isolation, and which is the current reality. And then we would be discussing phenotyping crops for stress combinations, particularly, and how the phenotyping is assisting to genomic assist breeding. Two major approaches which are being followed by different scientists are forward and reverse approach that I will discuss in brief, and then a few take-home messages, and finally I would conclude my presentation. Now, climate change and global food security, that's what is already discussed by Dr. Anil, previous speaker in this workshop. And then I would just briefly point out these important things. Is reaching towards 10 billion, so it's continuously increasing, and that's why demand for food crops is also growing very fast. On the other hand, rising global temperature with higher increased frequency streams like temperature, flooding, drought, forest fires, and they will decrease agree up to 50%. Now, displacement of thousands of coastal residents by sea level rise would further increase the pressure on global food security. Temperature and CO2 both are the prominent factors of climate change, and particularly after post-industrial era, particularly after 1960, we can see that rise in CO2 and temperature is very high. I mean, the pace of rise is very high, and now it is considered as a reality. Very prominent indicators which were already mentioned by Dr. Nielsen in last presentation, like sinking ice sheets, glacial retreat, and decreased snow cover, rising sea level stream. So this was the video actually by NASA, which is showing that climate change impact, how it is affecting the whole globe. Actually, skip it for now, and we can just go. How when agriculture production is concerned. Sir, am I audible now?
Yes.
Hello. Okay, thank you.
Yes, sir,
So
you are
weather
audible.
extreme and okay. Weather extreme and agricultural production. So when we consider particularly agricultural production, then we have to consider these factors which are directly affecting agricultural production worldwide, like spikes at regional level, short and long drought spells, limiting water resources, and irregular rainfall patterns. So these are particularly affecting agricultural production worldwide. And if we just summarize these factors, the heat and drought stress comes as factors which are affecting global food production. So this to summarize different events that happened across the globe in last 4 decades, and it is basically focused on heat, either heat wave or drought stress, and the causes that
You are visible now. Rajiv Nayan Bogura, आप visible हैं इस समय.
Yes, sir. But I'm not able to see how it is getting disconnected actually.
No problem. We are too much dependent on these technical things, na?
Yeah. Yeah,
So
certainly.
there is no fault of anyone in this. You please continue.
Okay. So I don't know from where I need to continue again.
Well, I
I'll
think
just
you said.
summarize. Okay. Can I start from this?
Where you left.
Is this slide visible, sir?
Yeah, it is visible.
Okay. So I will just go ahead from this slide only.
Yeah.
Okay. So this slide particularly summarize how water stress condition will be increasing in future, and as water stress condition will be increasing, the impact of temperature will be higher on a global scale. So this red area is showing probability of yield losses, and more red area shows more yield loss. And here also the red area shows high water deficient regions, and particularly if we see Indian region, it is very vulnerable for both heat and drought stress. Now, how phenotype and phenotyping is related to this workshop. Now I will try to connect the phenotyping portion. Plants are intrinsically related to their environment, and observed phenotypes are a direct product of genotype and environment. So this is the whole concept actually, that whenever we are discussing about phenotype, it is the product of genotype into environment. So most of the effort when we are doing to improve the genotype, and if we are not considering environment at the same time, then the probability of success is very low, because whenever you improve a genotype, we can do that for a particular environment, but as soon as any environmental factor is changing, the behavior of genotype will also change. So unlike a genotype, the phenotype of an organism can be described at many levels, from a specific molecule to dynamic molecule, metabolic network, to complex cellular developmental and physiological systems. Now, examples of phenotypic parameters for here, just for an example, like gross morphological measures like cell size, plant height, yield, dynamic measures such as metabolism, nutrient uptake, and physiological processes, and molecular measures such as transcript profiles. So all these are considered as a phenotype depending on the area we are looking into. Now, if we consider phenotyping research in the past and now the current status, so very spectacular advances in next generation DNA sequences have allowed genomic data to accumulate very rapidly. So initially in this graph, you can see that genomic data, sorry, can you see my presentation? Am I audible?
Yes, we can see.
Okay. So here in this graph in the right panel, we can see that development in the area of genomics and other proteomics were rising early in the 90s, while the phenomics was not getting progress in a similar way. But suddenly after 2009, or you can say after 2010, a sharp increase was seen in the area of phenomics. So this is what is actually I want to highlight, that most of the developments we were doing in the area of genomics were not getting translated into a proper successful strategy. Now it was believed that unless we describe a phenotype in a very right way, the success of getting genomics done is not at par. So we have to go in parallel with genotyping and phenotyping, and the accuracy level in genotyping should be achieved similar way in the area of phenotyping. So in the current decade particularly, we have achieved a number of papers in the area of phenotyping, and we are continuously achieving kind of development in the area of phenotyping. Here we can see that earlier, if you consider phenotyping, then it was mostly related to the yield and yield components, or mostly simple physiological processes, or just the grain weight or growth rate. All these things were getting considered, and the accuracy level was not at par with what we are expecting from the genomics data. Now, in the recent time, we have included imaging analysis techniques in phenotyping, which has made it very high throughput and very fast and accurate. Particularly in this slide, we can see that these are just wavelengths, different wavelengths, and different wavelengths are being used in different kind of instruments just to predict the changes, phenotypic changes in the plant. Like the early 500 to 700 nanometer wavelength is being used to analyze chlorophyll content, and then from 700 to somewhere 1400 is near infrared region, which is being used for vegetation health, which is basically reflectance based instruments, and then from 1400 to 2500 or 3000 is short wave infrared, which is being used for detecting water content, protein, lignin, and cellulose. So now these are the instruments which are basically using these particular wavelengths, and then they are able to tell us about the plant health in a very non-invasive way. So that's what is being used in high throughput phenotyping platform, and that is how we are now achieving the same rate of pace which we are going in the area of genomics. So precise phenotyping, when we are discussing about precise phenotyping, then we need to consider these important factors, and it's not just some trait which is visible and then we call it as, okay, we have done the phenotyping of the plants. It's not like that. So whenever we are developing climate resilient crops, we need to see what aspect actually we are considering. Like when we say this plant is tolerant in a particular condition, then there are three major strategies a plant is adopting. One is escape, another one is avoidance, and the third one is tolerance. And I particularly say that as true tolerance. Now we need to understand it. We need to understand this very clearly, that when I am saying escape, like in the case of heat or drought, escape is what plant is actually planning to escape the stress itself. Like for example, early morning flowering in some rice genotype, escape high temperature during the hot day hours. So they flower very early in the morning, like 6 a.m. or before that, so that the anthesis process is not exposed to high temperatures. Similarly, for the drought, flowering is being completed before the actual stress condition comes, or plant is just waiting to stress condition to go and then it is starting flowering, or any other important physiological process. So they are basically being smart to escape the stress condition itself. So we see the survival of the plant, but the strategy is not tolerance itself, but it is the escape strategy. Plant is not facing stress itself. Now another strategy is avoidance, where stress is coming and plant has a strategy to cope up with that stress condition, such as transpirational cooling under high temperature stress, or high root length in the under drought condition. So what is transpiration cooling? That the plant is absorbing water, right? And then by evaporative cooling, it is cooling down its canopy temperature. So if we see even the air temperature is 40 or 42 degrees centigrade, plant can maintain its tissue and canopy temperature well below the optimum limit, that is 23 to 24 degrees centigrade. So actually at tissue level, plant is not facing that temperature or stress because of this avoidance mechanism, right? And then comes to tolerance, where plant is genetically adapted to that stress at cellular level with the high expression of heat shock proteins or dehydrins or some kind of antioxidant enzymes which are continuously working to make the survival of plant in that particular stress condition. Now what happened actually, or what we could not include in our previous studies, is the stress combination, how plant is getting failed with these two major strategies, which we call as escape and avoidance. So whenever stress are coming in combination, these two strategies are not working properly, and the plant only which is truly tolerant to that particular stress condition can survive. So this is what I will try to emphasize, that whenever we are trying to characterize our plant phenotypically, we need to see which trait we are looking and in which category that trait comes in. Either it is in escape, or it is in avoidance, or it is actually related to true tolerance, because both escape and avoidance is something which will change as your environment changes. And I would like to say that most of the phenotyping which is done in previous decades was mostly related to escape and avoidance mechanisms. Now, stress, we know that anything which is going beyond optimum condition and affecting growth is stress. And in previously, as I have discussed, that most of the stress we have studies were in isolated conditions in a single environment. And that's why we have achieved very limited success in terms of crop yield under adverse conditions. So when we say stress combinations, it was mentioned by Dr. Anil sir in last presentation also, that those stresses can come simultaneously or sequentially or intermittently in one crop life cycle. So here are different possible combination, which is mentioned by Mittler and Blumward in their annual review paper. And these are the possible combinations they have studied, but there are still more possible combinations which we should look into. And then plant response to combination stress is entirely different than what we compare its response under an isolated condition. Okay, so when we say simultaneous, sequential, and intermittent, I have just put some examples like simultaneous occurrence may be heat and drought, or CO2 with temperature, or salinity with drought. In sequential, it may be flood to drought or flood to salinity, and in intermittent, drought may be followed by temperature or flooding at different time points in a life cycle. So these are possible stress combinations which we have to look into, because this is not the time when we say that, okay, plant is facing heat stress or plant is facing salinity and other stresses are not there, particularly if you are conducting your research in field condition. So it is impossible that plant is facing one stress at a time in changing environment. Now, these are obviously non-negative stress combinations, and I would be just focusing on heat and drought because that is the most of the time was the part of my research in last few years. If we say the current status of combination stresses, we can clearly say that even heat and drought has covered very less number of papers published, although a significant number has risen in last 5 years, because this review was published in 2015 by our group. And heat and drought was having only 2 studies at that time under natural field condition, right? So in rice, and if we consider ECO2, elevated CO2 with heat and drought, which is very obvious combinations in current and future climate, there were no study at all. So this is how I want to emphasize that where are we lacking in the area of phenotyping particularly, and when we consider a combination of different stresses. Now, some few intriguing questions before we go ahead, like how 2 or more stresses interact and how frequently these stresses combine in natural environment, how do plant perceive more than one stress at a time, and does plant response change under co-occurrence of 2 or more stresses as compared to a single stress, and can plant possess tolerance to multiple abiotic stresses? So I think these are very interesting questions we should ask to ourselves whenever we are working in this area. Just for example, what we were doing before and what is not known now is like impact of heat and impact of drought. Now, if you see impact of heat stress was majorly on sphagnum fertility, while impact of drought was mostly on photosynthesis, right? But when we combine these 2 stresses, and we actually don't know how plant is going to behave to combination of these 2 stresses, at germination, vegetative, reproductive, seed set, and grain filling stages. So these are the areas actually we have to see, or we have to explore, that whenever we are considering any sensitive stage or less sensitive stage, how plant is going to behave at the occurrence or co-occurrence of these 2 stresses or any combination of 2 or more stresses. Now, I will just skip this slide. It is just to re-emphasize how or why should we consider stress combination. So it's not simply additive, that's what is known. And in this slide, we can see these are the transcript and metabolites which are studied under heat and drought stress condition. And particularly, I want to emphasize this point where around 700 transcripts and 5 metabolites were very unique to the combination, and they were not there under heat or drought conditions in isolated condition. So it just shows that when stresses are coming in combination, different mechanisms are getting activated in the plant, and the response is based on those particular strategies. This particular interesting example is just to highlight how avoidance mechanism is not getting affected when stress combination is coming. So if you see this particular panel, when heat is coming, plant will open its stomata that will allow transpirational cooling, and that will just avoid thermal damage. On the other hand, if you consider drought, then plant will just close its stomata, that will decrease the transpiration rate, and that will improve the water use efficiency, and plant will again ensure its survival. But when heat and drought comes together, then we don't know what plant is going to do with its stomata, because plant is now confused. What is the result? That plant tissue temperature will increase, and it will again face dehydration stress, because there is no avoidance mechanism both for drought and heat stress, and plant survival is compromised. So this is very important to see that what can we do here, how can we help plant here, particularly at this question mark, so that we can make sure that whenever these two stresses are coming together, plant can ensure its survival. Now I would just highlight another combinations, because this is very important. Whenever we are talking about phenotyping, we have to talk about environment, and whenever we are talking about environment, it's not just one factor. It is not drought, it is not temperature, it is not light, it is not humidity. It is a cumulative or accumulation of all these factors working dynamically within a particular region or environment, right? So if we consider temperature, light, and humidity, so whenever we consider the effect of temperature, actually, it is not just the temperature which is affecting plants. So if we see heat stress impact on crop is largely regulated by air moisture content, right? So temperature stress is largely dependent on the moisture content of air. Now, high solar radiation and low moisture content can increase vapor pressure deficit, which support transpirational cooling during hot hours. So if we say that, okay, plant is efficiently doing avoidance mechanism, that depends on the environment where solar radiation should be high, moisture content should be low, which will increase vapor pressure deficit, and then only plant is able to do transpirational cooling. So there is one very interesting, sir, am I audible? Is it going fine?
Yes, sir, it's going fine, sir.
You are perfectly audible.
Okay. So I would just put a very interesting case here, that somebody from African Rice Center came to Erie when I was doing postdoc there, and they came with the genotype, rice genotype, and they have claimed that this genotype can survive up to 45 degrees centigrade temperature, and it was doing flowering and everything so fine. So we were looking for a tolerant genotype, because N22 is the N22 is the only tolerant genotype recognized globally for rice, and due to its poor combining ability, very poor agronomic characters, it is a very poor choice for breeders to do kind of crossing job. So we were definitely looking for that. So we told that scientist, okay, we will try your genotype here, and we just grew that genotype in Philippines, and that catastrophically failed. It was not able to actually survive in that environment, and very high spikelet sterility rate was observed. So that scientist was very confused that why this happened, and then we tried to explain the case with him that in Africa, he was living in very dry environment, right? So even with high kind of temperature, air temperature, there was a high vapor pressure deficit, which were allowing thermal cooling at the canopy level. While Philippines is a very humid area where very low vapor pressure deficit was not allowing any kind of transpirational cooling. And that's why the temperature effect was very high on the plant. So this was very interesting example that if we are not considering environmental factors, then we get confused actually with our results. So in one environment, one genotype is very successful, and in another environment, it is totally failed, because we have not considered different factors which can affect actually the crop performance. So whenever we are considering temperature, we have to consider the light and humidity levels. So in this review, I have just tried to summarize how these factors actually affect different processes in the plant. So temperature, whenever we are talking about temperature, within temperature, we see the variation. Like if we go to temperature extremes, then it may be cold temperature, it may be heat temperature. Then within temperature, there may be seasonal variation, there may be diurnal variation. And then along with all these variations in temperature can interact with light, and then that will affect tissue temperature, nutrient absorption, and stomatal conductivity. Similarly, temperature with all these variations may interact with humidity, and that will affect germination, photosynthesis, flowering time, and dormancy. Now, within temperature, if we say night temperature is having a different impact on the plant, where it is basically affecting sourcing regulation, which is affecting seed development and quality, while the day temperature is affecting reproductive development, and that is ultimately deciding the flowering and seed set process. And then interestingly, the difference between these two, which we call as amplitude, is perceived by plant in a very different way. That signaling is still not known, and that is affecting carbon use efficiency. Here we can see that even just with the temperature, if you are increasing your temperature in a growth chamber, making it 40 degree, making it 38 degree, and then saying that, okay, my plant is behaving very nicely, now it is a tolerant genotype I have discovered, it's not going to work whenever you are growing your plant under natural field conditions. So this is the reason, basically, because we have not included these factors in a dynamic environment, and then how these factors are changing plant behavior actually. So these are obvious combinations, and I would be just focusing on these two stresses, like high day temperature with drought stress, high night temperature with drought stress. This was what we were doing in past few years. So we have discovered few traits for stress combinations, particularly for heat and drought, and as much as success I could achieve in past few years, I will just try to highlight in next few slides. So we have seen both in C4, and rice crop, it is reported that photosynthesis, stomatal conductance, and transpiration, these are basically gas exchange traits. And we have seen additive effect of heat and drought stress combination. So even if plant is able to survive under heat stress or drought stress in isolated condition, we could see that gas exchange traits are just getting affected severely whenever heat and drought combinations were happening. But if you can see here, particularly stomatal conductance, like their behavior, its behavior is very different when heat and drought is coming in combination. So it's not kind of in the rule book that whenever heat and drought is coming, then the effect will be additive or not additive. So it depends on the genotype we are working on. It depends on the environment where we are working, and then it depends how these two stresses are occurring either together or just following one after another. This was a nighttime stomatal conductance and transpiration. This was my major work actually in Philippines, and we are just working on a very big publication actually out of this study, where we have accumulated around 7 years data on these traits, where we have seen that in this slide you can see that this is stomatal conductance and this is transpiration, and this is happening under high night temperature, and this is same stomatal conductance and transpiration under drought condition in the night time. So very significant amount of water is being released by plants in the night, right? So these traits were never included actually in the tolerance kind of breeding for heat temperature or drought stress tolerance. So we could see that these traits are very, very significantly working in the plant, and at the night time actually plant is designing its kind of survival strategies, right? And that's where actually we need to see how plant is doing it, not like we should impose our kind of strategies on the plant, but we have to look very precisely how plant is doing it under the natural environment, and that's where we explore genetic diversity, where one plant may be able to cope up with this stress with its different strategy, while other plants cannot do that. Similarly, like I stated in my previous slide, how solar radiation is affecting BPD. and how relative humidity is affecting BPD, and then both are allowing kind of transpirational cooling. And in another case, we just summarize that if we can reduce nighttime transpiration under drought condition and even under heat stress condition, that could be very potential trait for both drought and temperature stress combinations. Now, instrumental closure during daytime can cost heavily for carbon fixation through photosynthesis. So, genetic ability to lose less water with tightly regulated instrumental behavior could be a key to identify very promising and tolerant genotypes. So, these genotypes is basically they are trying to keep their stromata closed during the night time to save water, and in the daytime, in early day hours, they can open their stromata, do most of the photosynthesis job, and then they shut down their stromata to save water. So stromatal sensitivity is a very key trait actually we are looking into. Now, due to stromatal closure during day hours, efficient uptake of soil nutrient during night time could ensure survival under heat and drought combination. So now it should be a balanced distribution of these activities during day and night time, and we are continuously studying this pattern, how plant is behaving with these traits in the night and in the early morning, and then late in the hot hours where drought and heat stress can combine and affect plant's ability. Another trait which we combined with the nighttime transpiration is leaf thickness. So leaf thickness is defined as increased dry weight per unit area, which we call as specific leaf weight. And it has been reported earlier that thicker leaves mean a greater thermal mass, which increases thermal stability. So it basically helps in both way actually, where drought is happening or temperature is coming in high. So thermal stability can be achieved with thicker leaves. So genotypes which are having thicker leaves, they can avoid heat-induced damage during daytime hot hours, and they could transpire more water actually during the critical hour of photosynthesis, because they have preserved more water at the tissue level. So both reduced nighttime transpiration and thicker leaves could be potential targets for breeders for heat and drought combination stress tolerance. Another very important trait which is being highlighted is nighttime respiration, and which we call mostly as maintenance respiration. So respiration is the process by which plant actually increased or produce energy for the growth and development, and that respiration is broadly categorized in growth respiration and maintenance respiration. So growth respiration is whenever the plant is in active growth phase, up to heading stage in rice. So most of the respiration is happening for growth purposes, right? Active growth purposes, while maintenance respiration is getting higher during the later period where reproductive development is happening and plant is not growing further and it has to just maintain the biomass, right? So at this time when stress is getting imposed on the plant, the cost of maintenance is increasing by repair, uptake, and transport of nutrients, and that's where the most of the accumulate actually, the photoassimilate is being utilized in this respiration process, and that is directly affecting our yield and productivity. Now we can see that more than 60 to 70% of carbon fixed in photosynthesis is normally burnt in respiration. So if this proportion is increasing by 10 to 20%, then definitely we are not going to get yield as per our expectations. Now stress-induced changes in respiration affect the carbon balance, and higher respiration under stress could reduce biomass, grain weight, and grain quality traits. We have explored genetic diversity for respiration, and we have tried to identify plants which can maintain low respiration even under the stress conditions. So this is very key trait which breeders have not looked, actually even the physiologists have not looked earlier, and this is very potential trait because this 60 to 70% loss in respiration and even more under stress condition can be reduced. And if we select plants which are very smart in using their energy budget, they can be our target actually to design climate resilient crops. So here we can see that even if we are considering respiration as a trait, then where should we look and how should we take respiration? That is very important, right? So it's not just we are going into the field, we are taking respiration rate and we should be satisfied that we have taken this rate and we have characterized our plant. So that's not the case. Like in rice, when we did respiration measurements throughout the growth cycle, we could see that this respiration was actually going higher during post-flowering period. Like I have said, that growth respiration, and I should mention here that growth respiration is mostly independent of temperature. So even if temperature is increasing, that's not affecting the rate of growth respiration. But any kind of stress can increase the maintenance respiration, and that's where we can see here that whenever stress is high, this maintenance cost was increased, and during the post-flowering period. So here we see that temperature stress was given at pre-flowering stage also. You can say it covers from early vegetative to late vegetative stage, and we have seen increase in maintenance respiration, but that was not very high. But here it was very significantly high. So we have taken 212 genotypes actually to just conclude this, and this was very solid evidence that maintenance cost is increasing during post-flowering phase. A similar observation was done for drought and flooded condition, and we could see that at post-flowering stage, the cost of maintenance is very high, and that is increasing overall the respiration cost. Now, when we compared it with photosynthesis, we could see for different genotypes, and we saw that in post-flowering phase, effect on photosynthesis was not very high. So we could not see any significant change in the process of photosynthesis, although it was decreasing gradually as we moved towards grain filling stage. But cost of respiration, if you can see, it clearly under 29 degrees centigrade, the cost of respiration was high even after 25 days post-flowering. So even during the grain filling period, where a significant amount of photosynthesis was not happening, respiration maintenance respiration was still going high, which was actually costing the accumulated photoassimilate in the grain and reducing overall grain weight and quality. Now, even we have taken the time actually, which time should be considered for respiration measurement. So this was done for photosynthesis and respiration. In photosynthesis, early day hours were the correct time window to take photosynthetic measurements. And then for respiration measurements, mostly we have found that it's around 1 or 2 in the night actually. 1 or 2 a.m. in the night was the perfect time window to measure the respiration. And that's why in Erie, I think, for almost 2 years, every night I used to be in field from 11 to early morning 3 or 3:30 a.m. So these were the respiration measurements we were doing actually. And this time window was found perfect, and that's why I think people are hesitant of taking up these traits actually. Now even respiration is directly dependent on the temperature, and we have seen that if you increase the leaf temperature, means that is a direct consequence of increasing air temperature. So if air temperature is high, like if you are in a region which is very hot, then your respiration rate will naturally come higher, right? So if you are comparing your results, one study in Philippines where temperature is around 25 to 26 throughout the year, and then we are comparing it with some North India, summers Kharif region, then that difference due to regional level is just due to temperature actually, that temperature difference. So we need to rule out these kind of changes which are coming due to natural environmental differences, and that's how we have just concluded it in our paper. Another tool we were using to characterize genotypes was thermal imaging, and this is very actually popular tool now to characterize genotypes. And this is, as I mentioned, this is particularly working on avoidance mechanism where thermal cooling at canopy level is being recognized as a strategy of plant to survive under drought and heat temperature conditions. So here we can see that these regions, blue regions, are cooler canopies, and they are the hotter canopies. And just at a thermal imaging, we could see that, okay, these plants are having a kind of a significant avoidance mechanism under stress conditions. This was done in our Erie experiment. We were having thermal rings there, and we used to expose the plant to higher night temperature, and we could characterize different plants which were having that thermal avoidance strategy. based on their cooler canopies. Relative water content is very important trait. I would not go in detail with this trait, but this is very potential trait we have found for heat and drought stress condition. Another was mineral profile we have published actually, and we have seen that ability of a plant to accumulate mineral components from the soil decides its tolerance level. So we have to see how plant is accumulating mineral components from the soil, and it was directly related to its tolerance level to any kind of stress actually. So that is just like we take multivitamins and minerals just to improve our immunity. They are directly connected to the immunity of the plant, and any genotypes which can accumulate more and more minerals under stress condition is definitely going to be tolerant under that particular stress condition. Similarly, antioxidant machinery are kind of a core zone of defense process, and we could see that anyway, any kind of stress is going to increase antioxidant mechanism. And as far as our study was concerned, we could see the role of superoxide dismutase and peroxidase mostly under stress condition, and we could consistently see that low ascorbate peroxidase and catalase activity was found in our studies, and that was suggesting the role of salicylic acid mediated signaling under stress condition, which is definitely a topic of interest for molecular biologists. See how this salicylic acid is basically playing role under heat stress tolerance, drought stress tolerance, and then we need to explore how it is working under the combination of heat and drought stress tolerance. Another important aspect of my postdoc work and the work I am doing now here was forcing dynamics, and we could see two key enzymes. One is invertase and another one is sucrose synthase, which were very crucial to decide zinc strength. And we could see that under stress conditions, these enzymes are deciding genotype's ability to survive under stress conditions. Here in this particular slide, I have seen that for high night temperature, but now we are characterizing plants for these traits for high night temperature and drought combination and drought with salinity. So here we can see again. So if you say that, okay, we are looking into starch synthesis, right, process, and in starch synthesis we have seen, okay, in leaf this starch synthesis was decreased, right? Here we have compared in source and grain, and we could see that in grain actually where we need starch, that starch synthesis process was not affected. So in the grain we would say, okay, this starch synthesis was not affected because this starch synthesis enzyme is working fine. And here in the leaf it is decreased, but actual impact of stress, the process of starch accumulation in the grain was seen with the activity of this cell wall invertase enzyme and sucrose synthase. So even if this particular enzyme was not getting affected due to poor activity of cell wall invertase and sucrose synthase, there was not enough substrate for this enzyme, and that's why this starch synthesis was affected, and this has reduced the grain weight and actually the rice grain quality parameters. Further to that, we could see that accumulated starch content was decreased during grain filling period, and that was due to the higher activity of this enzyme, which is called alpha amylase. And because during the later period of grain filling, most of the enzymes are not active due to desiccation process, because water is getting taken out from the grain for the maturity, and that's how this particular enzyme becomes very significant, because the plant is facing stress at that stage. Then energy requirement is coming from breakdown of this starch, which is already accumulated in the grain, and then that starch is broken down for energy synthesis and maintenance, and that's why this enzyme activity was seen higher under stress conditions. So we have to see in a very holistic way that even if we are considering some enzymes during the grain filling period, then which enzyme in which tissue at which stage we have to see, conclude, okay, this is how or this is where the process is getting affected. So even if we are having kind of genotype with a very high sucrose synthase activity or genes expressed for that, we need to see how cell wall invertase is actually able to unload the sugar, and that sugar is not getting accumulated in the rachis part, which was the case in our study, that most of the panicle sugar was remaining there in the rachis, and that was not getting into the spikelet because the cell wall invertase was not able to transfer that or unload that sugar into the developing grain. Now, apart from the above ground traits, these are the below ground traits. Actually, we are focusing particularly with Winrhizo study, and but we are looking into is root surface area, root length, root angle, root hydraulic conductance, and then rhizospheric microbes, their role actually. And it is actually known that how the microbes can boost antioxidant machinery, they can help in maintaining cell turgor, they can help in nutrient supply, and they can even help in temperature regulation by increasing root surface area and then providing more water to the canopy. So these traits are equally important and they are not at all explored actually. So these are just summarization of the traits we are looking for day and night, and then above ground and below ground. So I have just explained it in my previous slide, but that's how I am trying to say that it's not just one trait or a couple of traits which are going to help. We need to see how conditions are changing, how environment is changing, and then we have to select or pick the traits which are very relevant in that particular environment condition. This is one example where we could successfully characterize multiple abiotic stress tolerance, and this particular thing was done with gamma radiated IR64 mutant lines, and we have characterized these lines phenotypically, and we could see under salinity and drought stress conditions, these lines were very potential, and we could see particularly this line M3 and M2, they were working very nicely. Actually, they were performing, and this was just applied for as Indian patent application. So I don't know the current status of this, but I have personally seen the performance of these genotypes. And yes, if we can try to phenotype them correctly, we can find genotypes with multiple abiotic stress tolerance. This is just a preliminary kind of example, but we can certainly go ahead with these kind of studies, and then we can see that potential genotypes can be found out with different strategies along with genomics strategies which we are actually doing. Now, how phenotyping is basically trying to help in genomics-assisted breeding. Now, establishing the connection between genotype and phenotype is currently one of the most significant challenges facing modern plant biology. The limiting factor of quantifying phenotypes, particularly in the context of agriculture and native plant population, has been the lack of phenotypic information of scale, density, and accuracy comparable to DNA sequencing data. So that was the major problem in phenotypic area, that we were not having repeatability and accuracy. Now we have achieved that, right? So we have to see actually that is very complex biology, where we have to consider environmental factors, we have to consider the instruments we are doing, we have to consider the plant background, we have to consider different growth stages like that. So that is what we are trying to do. And this is one of the study which was published by our group in plant physiology. And this was a huge phenotyping exercise led by me. I was leading the phenotyping part of it. And we have phenotyped 35 traits, mostly related to root morphology and anatomy. And that was covering 45,000 root scanning images by Winrhizo and nearly 25,000 cross taxons from the root shoot junction. Now that was then carried out for a genome-wide association analysis, and we could see priority genes, candidate genes, number of under control, water deficit stress, and then plasticity, means how plant is changing under controlled water deficit conditions. So that kind of plasticity in plant. So we have seen around 233 genes for that, and now we are trying to just select a few of them for the validation purpose, and that study is still going on. And then we are trying to use those genes to improve genotype, and they are either being suggested for the breeders or molecular biologists to incorporate in elite cultivars. These were the two strategies I was trying to explain actually, and this was reviewed by me in one of the review paper. Two major approaches which students should look into. One is the reverse genetic approach, and one is the forward genetic approach, where we should take reverse and where we should try forward genetic approach. That I will try to explain. Now, if we have a elite genotype, we have a potential genotype already with us, then we can just go with recombinant DNA technology, where insertion, construction, and overexpression of genes, or RNAi technology or genome editing can be used to modify our gene of interest. Or we can just go to site-directed mutagenesis, and then mutation in gene of interest will modify our gene of interest. Now we need to precisely phenotype it, and then screening them for multiple abiotic stress tolerance. which will lead to identification and characterization of multiple abiotic tolerant lines. On the other hand, if we see the forward genetic approaches, then we need to have a diverse genetic panel, and then if genetic diversity is very high, we can directly enter into the precise phenotyping of those genotypes for multiple abiotic stresses, whatever our interest is. Like we are doing it for heat and drought stress, and then we can identify the donor lines. Like for heat stress, we have already identified Nerica L44, which is published already in Physiology of Plantarum, and it is being used by breeders in Erie and at IRI. Now we can confirm the gene or locus governing that multiple abiotic stress tolerance using that donor line with any kind of elite or sensitive line. If we have low genetic diversity in our panel, then we can go for mutagenesis, like we have done that IR64 mutant lines, and then random mutations will be generated, and then that large population can be phenotyped for different kind of stresses or combination of different stresses, which will lead to identification and characterization of multiple abiotic stress tolerant mutant lines. Now, the traits or genes will be introgressed into the elite cultivars through breeding or biotechnological approaches, and finally we will get the climate resilient crops. So these are the roadmaps actually being utilized by scientists across the globe. So what are the challenges particularly? So I will not cover the challenges which are covering in genomic sciences, because the experts are already knowing that, and our eminent speakers has already mentioned those things, but I will just highlight the challenges which we are facing in the phenotyping area. So regarding nocturnal gas exchange traits is a big challenge. I have done that, and I know it's not an easy job. Then understanding role of below ground root traits to multiple stress combinations, it is not at all explored, and it is a major bottleneck actually from the physiologist side, which we need to tell breeders how important are those traits, and they should include them if they are trying to develop climate resilient cultivars. Then plant response to mixture of coinciding stresses conditions like that require very precise phenotyping, and our observations should be very, very precise. And then it is very important to understand the connection between various environmental factors, like I have mentioned that temperature with humidity and light, and drought with moisture content, and the soil properties. Then those environmental factors, how they are affecting plant traits at critical growth stages. What are the opportunities we have now in this particular decade? Now we have high throughput phenotypic plate. We have high throughput phenotyping platform, which are excellent for precise phenotyping for plants under controlled environment. And even we have now, even if your genotype is heat tolerant, even if your genotype is drought tolerant, that may not work in a particular environment where heat and drought are interacting, or the environmental conditions are different from the environment they were developed. Now, precise phenotyping for potential trait is very effective under multiple stress combination. So we have to include phenotypic exercise along with the genotyping and all the molecular characterization. We should consider phenotyping as a kind of prerequisite tool to follow before we go in detail and then see the molecular processes in the plant. Finally, incorporation of robotics, artificial intelligence could minimize the human errors, and observation would be improved, and the pace of phenotyping will be high. Now, genomic-assisted breeding requires well-characterized plant phenome. So, plant phenome is just basically plant phenotyping when we know a trait of a phenotypic trait and then its associated gene. Then we call it as phenome or phenomics, right? So, all the genomic-assisted breeding require well-characterized trait and its associated gene. So when we have this information, then only we can think that we will achieve required success in the field of developing crops for climate resilience. So I will just conclude now. Effect of gaining higher yield under stress environment in past has achieved limited success, and one of the major reasons for this was lack of phenotypic characterization and understanding the plant response to complex interaction of multiple stresses. Then potential traits effective under stress combinations could help in identifying genotypes with tolerance to multiple abiotic stresses. And finally, phenotypic traits need to connect with genomics to identify associated potential genes that could be introgressed into the elite cultivar to maintain higher yield even under extreme environmental conditions. So these were two facilities. The above one was being used by me during my postdoc at ERI, and a similar kind of facility I have developed in IRI, and these were the environmental facility at IRI we were using. And now we have a very high throughput phenotypic platform developed at IRI, which is being provided, I think, for all the scientists working in this area. I would like to acknowledge our director here at Center of Advanced Studies on Climate Change for providing all the facilities for my research. This grant was utilized by me for most of the research I have shown in previous slides. And I would like to acknowledge my active collaborators like Dr. Krishna Jagdish from Kansas State University, USA, Dr. Ashwani Parikh from JNU, and Dr. C. Viswanathan and Professor Madan Pal from IRI in New Delhi. Thank you so much. I think everybody was listening.
Thank you very much.
Thank you so much.
We are
So I would
waiting.
like to open for questions.
I am very much thankful to you because you have started from the very beginning, just like a teacher, and you have covered almost 10 to 15 slides for the basics of plant abiotic stresses. I am very much sure that the students will be benefited from your lecture. Then you have advanced further in the research area, and finally you have come to the conclusion. So you are a very excellent teacher, Dr. Bahuguna, and I am again very thankful for being very gentle because of
for the nice introduction. So, and also thank you for giving me an opportunity to be part of this workshop. And, so the topic of presentation is rice genomics for biotic stress tolerance. And again, to introduce myself, I am Dr. Tangasuja Srinivasan from Agriculture Biotechnology Lab, Center for Climate Change Studies, Satyabama Institute of Science and Technology. And for people who do not know where Satyabama Institute of Science and Technology is, it's in Chennai, Tamil Nadu. And this is a beautiful campus. And in Satyabama, we are in the International Research Center where our Agriculture Biotechnology Lab is located. So, and what we do in our Agriculture Biotechnology Lab, Satyabama Institute, is we are mainly focused on biotic and abiotic stress tolerance mechanism of crop plants. And for this, we are using rice as one of our model system. In rice, we are trying to understand the mechanism of rice resistance to plant and leaf hoppers. Actually, our previous studies have proved that plant hopper assemblages, when fed on a resistant rice variety, is able to perform better than when compared to individual colonies. So this made us to frame a hypothesis that the interaction between plant hopper assemblages may be facilitated by some mechanism in rice, and we hypothetically say that the mechanism could be the phloem sap of rice. So in this study, we are basically trying to extract the phloem sap of rice and trying to reveal the induced defense response components in the phloem sap of rice. And we are in the process of we are moving in forward in this study. Apart from not exposed to a single stress factor, it is like a combination of different stress conditions along with the environmental factors. So, we actually do not know how plants actually cope with all these multiple stress factors under stress condition. And in this study, we are mainly looking into the regulators, that is the molecular regulators that are helping the plants to give out the tailored defense response. And we are looking into the NTRs and MAP kinases and WRKY like transcription factors in this study. Apart from that, we are also looking into the insect adaptation mechanism to resistant host plants as well as to different insecticides. And for this, we are using cigarette beetle as our host model insect, where we are trying to reveal the different detoxifying enzymes as well as the sequestration pattern in the insect. Along with that, we are also looking into the gut microbe of the insect, like whether the gut microbe has a role in helping the insect to adapt to a particular host plant or to a particular insecticide. So this is in brief about what we are doing at Satyabama. Now, getting into the talk, I can say that I have made the presentation as quite simple, so that it is like I mean it's useful for beginners and researchers. So like we would see like about a little bit about the rice genome and what advancement have been made in rice genome sequencing, how this rice genomics has been involved in identifying biotic stress related genes, and how it could be involved in identifying the crosstalk in plant defense signaling mechanism, and the final summary. So starting with the basics, and the first is like what is genomics? Genomics is nothing but the entire genetic information of an organism, and the word was actually coined by Thomas Roderick in 1986. And how far it is different from genetics, and how far genomics is different from genetics is nothing like genetics deals with the inheritance of genes, like inheritance of a particular gene or a QTL, which is located in a specific of a chromosome. Whereas in genomics, you study the entire genome organization, and you also address all the genes that are in the genome and how they are interacting with each other and how a response is mediated in an organism. All these things comes under genomics. And this genomics is like broadly categorized into three different types: structural genomics, comparative genomics, and functional genomics. In structural genomics, it's basically the structure of the genome organization, as the like it's comparatively easier to to do, like when compared to functional and structural comparative genomics. In structural genomics, you come out with the size of the genome, the number of genes in a organism, or the number of coding regions, number of non-coding regions, repeats, as well as no, I mean, all these things will be covered in structural genomics. Whereas in functional genomics, which is a bit complicated than structural genomics, where you try to address the function of the genes and how these genes are interacting with each other in mediating a response. And in functional genomics, mostly techniques like overexpressing of a particular gene or protein-protein interaction studies and gene silencing and gene knockdown techniques are commonly used. And the last one, that is comparative genomics, which is a bit complicated when compared to the other ones, where you directly compare the genome of one organism against that of the other. And why we do it? It is like mainly to know how an organism has evolved, or like in other words, like what is the evolutionary relationship with different species of an organism. And apart from evolutionary relationship, in case of comparative genomics, you will come to know about the differences and similarities within and between species. And also, like it's in comparative genomics, you come to know about the relationship of the genome organization and the function across various biological organisms strains. And yeah, now coming to the rice genome, I mean, rice was, as everyone knows, that rice is a model cropland for monocots, and it was the first sequenced crop genome after Arabidopsis. And the reason for choosing rice as the crop genome to be in monocots is that, like, rice was having a smaller genome size, as everyone knows, comparing to the other grass species, and it is deployed in nature where Q1 is equal to 24, and and it had a higher degree of collinearity also with other related grass species. Say, for example, perennial whitegrass. It had a higher degree of collinearity with the other grass species, and it also had a greater biodiversity and thing. Like the role of biodiversity is that, like many of the agronomically important traits, as well as biotic and abiotic stress-associated genes, were found to be in the gene pool, and this was an added advantage. And in case of Oryza, the genus Oryza, where you have two cultivated species, one is Oryza sativa, the other one is Oryza glaberrima, which is the African rice, and that it had 21 wild relatives. And these Oryza species are divided into 11 genotypes, 6 of which are diploid and 5 of which are polyploid. And the cultivated Oryza sativa, that is the Asian rice, belongs to the AA genome group. And this Oryza sativa is further divided into two major subspecies. That is the Oryza sativa subspecies Japonica, which is the sticky short grain rice, and the non-sticky long grain rice, which is the indica type. And this indica type has also further subgroups, for example, like Os type and Goro type and Aman type. And all these land races have been growing under extreme environmental conditions, and growing under the extreme environmental conditions have helped them to retain all their biotic and abiotic stress tolerance genes. Say, for example, the submergence tolerant gene SUB1 was identified from the Os type, and the phosphorus starvation gene, that is PSTOR, was again identified from the Os type, and our famous N22, the drought-tolerant variety, was again Os type cultivar. So, and again, as I was saying, that rice had a higher degree of collinearity with other related grass species, and comparative analysis of related grass species have shown a conserved synteny over a long-term evolutionary time frame, resulting in a concentric circular genome, where you see the gene orders to be, by the gene orders among different grass species. Say, for example, you can see the pigmentation associated gene signing to be in the same order in wheat, maize, and rice, and also the darkening gene in the same order in maize and in wheat, and again the ligules associated gene in wheat, maize, and rice. And also, I should say that availability of rice genome data had favored the, in identification of agronomically important traits in other grass species. Say, for example, powdery mildew associated trait in barley, malting quality in barley was identified based on comparative analysis of rice, and also apart from that, heading date in perennial ryegrass was based on the comparative analysis of rice. And now coming to the rice genome sequencing thing, there are four institutes which played a major role in sequencing the international rice genome by the Rice Genome Program, Japan, and the Beijing's Genomic Institute, China, and two private firms, Monsanto and Syngenta. And they had two sequencing techniques. That is, one is map-based sequencing, and the other one is short-term sequencing. And we know what is that in the next few slides. So coming to the milestones in rice genome sequencing, actually the sequencing of the rice genome was actually launched in very much earlier than the release of Arabidopsis data in 1997, with the collaboration of 10 different countries, and India being one of the partner in sequencing rice. And followed by that, in 2000 and 2001, Monsanto and Syngenta had a draft sequence of rice covering 95% of the rice genes were identified in the draft genome. And in the following year, in December 2002, International Rice Genome Sequencing Project completed its sequencing and submitted the high quality draft sequence by clone approach with less than one error per 10,000 base pairs, and it covered around 366 MB of the genome, corresponding to 92% of the rice genome. And subsequently, in 2003, the International Varieties Map Alignment Project was initiated with the aim of providing high-quality wild rice genome resources. And in 2004, International Rice Genome Sequencing Project produced a high-quality sequence of the entire rice genome with 99.9% accuracy. And in 2012, the revised version of Nippon-Beret genome was released. And in 2014, the 3K Rice Genome Project, which included the 3,000 rice accessions, which were from different countries, and the International Rice Research Institute Philippines, as well as the Beijing's Genomics Institute China, played an important role in the 3,000 rice accessions. And in 2017, Varieties glaberrima cultivars two of the Varieties glaberrima cultivars were sequenced and submitted. And in 2018, new reference assemblies were made available for 6 wild Varieties species with AA, B, and FF genotypes, and along with 2 domesticated Varieties sativa indica cultivar, that is IRA, which is the miracle rice of Asia, and N22 cultivars were made available. Now, coming to the sequencing technologies which were used in sequencing rice, one is the short-pen sequencing approach, which is basically like a faster technique when compared to map-based sequencing, where you have short fragments. You chop the genome into small DNA fragments, and you sequence each of the fragments, and then align them and come out with the sequence. But the problem associated, this was quite faster, but the problem associated with this is that it had many gaps, and the repeat sequences were not properly placed, and they were not able to predict how much is the length of the repeat sequence and how many repeats are present in that particular. These were the pitfalls associated with the rice genome that was available in 2000. And consequently, in 2002, the map-based sequencing, which was based on clone-by-clone approach, where each stretch of the sequence is anchored to the chromosome, forming the contact, and they were then individually sequenced. Although this technique took lots of time and labor, it was quite reliable and it was much more accurate. So it could be said that this particular thing had a reduced error rate of less than 1 per 10,000 base pair, and they were also able to cover many of the gaps using this technique. And as I was saying previously, the 10 different countries were participated in the International Rice Sequencing Project, and they were able to cover the 12 different chromosomes along with 2 centromere regions. And India was one of the members, covering the short term of chromosome 11. Today, like the numerous rice accessions have been successfully sequenced, and this is data of showing that the different rice accessions that have been successfully sequenced, and the data for all these accessions have been available online. Now, what is the significance of rice genome sequencing is that they were able to actually sequence the 370 MB of the India brain, and they cover 99% of the euchromatin, and this regions covered about 40,000. Like many of the genes have been economically important, and biotic and stress tolerance genes have been successfully cloned, and many of them have been partially characterized also. And it is like from the genome, it was predicted that one gene was found to be in every 9.9 kilobase, and but this was a quite lower density when compared to Arabidopsis, and the reason could be that it could be gene duplication events or gene insertion, which would have happened in the long course of evolution. Then also like comparing comparative analysis of Arabidopsis with that of rice showed that 71% of homology with Arabidopsis was, and 90% of the Arabidopsis had a putative homology with rice, and there was also evidences for organelle DNA transfer, and 2,859 unique genes that were specific to rice and other cereals were identified. And there's the applications of this rice genome sequencing is that it had fastened the marker-assisted selection as well as molecular reading techniques, and it had also helped us to understand the interaction mechanism between rice and also its biotic and abiotic stress factors. Apart from that, we have also been able to improve the nutritional value of rice and enhance the profit by improving the seed quality, resistance to pests and diseases, and plant hardiness. And it is also useful in identifying the plant stress response pathways and interactions. And obviously, it has hastened the development of genetic markers in rice as well as in other grass genomes. Now, coming to rice genomics in biotic stress, like using the rice genomic data, so far so many biotic stress associated genes have been identified almost on all the 12 different chromosomes of rice. And before getting into that, I would like to say briefly about the biotic stress mechanism in case of plants. I will not say that this is the specific mechanism in plants, like, but this is a broad category where you have the systemic acquired resistance and induced systemic resistance. Systemic acquired resistance is mainly mediated through the R gene, gene-to-gene interaction pattern, where there is an effector and the effector is identified by the receptor, and then comes the downstream. And actually, it is a broad range of resistant mechanism thing, and it is systemic in nature, and it mainly leads to the pathogenesis related gene expression thing. And here, salicylic acid is the predominant phytohormone, and induced systemic resistance, where we mainly involve non-pathogenic rhizobacteria, and it is mainly a priming response, where you prime the plant to be ready for a future attack. That is the main thing here. And here it is independent of salicylic acid, and here the main thing is jasmonic acid and ethylene, and it is independent of PR gene activation, and all our plant growth promoting all those rhizobacteria comes under this where they actually prime a plant for an or they give a warning signal for the plant for a future attack. So now coming to the genes and proteins that have been associated with disease resistance. So there are so many genes and proteins that have been identified. For example, in bacterial blight alone, 42 bacterial blight resistant genes conferring resistance to xanthomonas have been identified, and many of these genes have been found to be nucleotide binding site leucine-rich repeats and receptor kinases. And actually, the main function of the nucleotide binding site leucine-rich repeats is that the effector molecules, the effectors which are injected or which are delivered into the cell is being identified by these nucleotide binding site leucine-rich repeats, and the downstream signaling process is activated. And And in plants like this nucleotide binding site mucin which repeats, so many MPS have been identified, and even in case of not only for disease resistance but also yes, LLRs have been identified for insect resistance, especially with flowing feeding insects. And there have been so many resistant genes that have been identified for bacterial blight, and few of them are also resistive genes, of which the I mean, an interesting one is this XA30, which have been widely made in allied cultivars in India, which is like a mutation in the promoter binding site of promoter binding site of the TAL effector that thereby prevents the binding of the effector genes, and thereby sugar transportation is prevented or hindered, resulting in the hindrance of sugar effects to the bacterial cells, thereby the bacteria cannot survive. And there is an interesting mechanism with XA30. And apart from bacterial blight, in case of sheet blight, more than 200 QTLs associated with sheet blight resistance have been identified from mapping populations of rice, and 14 QTLs related to bacterial panicle blight resistance have been identified in rice. And in case of blast disease, like more than 350 QTLs have been identified on the 12 different chromosomes of rice, and of which 25 QTLs have been molecularly characterized. Like we have identified so many number of QTLs and different chromosomes of rice, and when when we ask like how many have actually a particular role, have only like few numbers. We could make it as like either 5 or 10 or like 20, and that is the one of the major pitfalls in this QTL identification and characterization. And this is a picture where you can see that all the 12 different chromosomes of rice are being clustered with the identified, so far identified disease resistance genes and QTLs. And this shows that how many different genes could have been identified from a same region using a different disease. For example, identifying the same region in blast as well as using bacterial blood. That is even possible. And this is again a picture showing the distribution of genes across the 12 different chromosomes. Where you can see that in chromosome number 3, we have a really less number of genes identified, whereas on chromosome 11 and 12, there was a really hike in the number of genes that have been identified so far. And in case of insect resistance, also the same case continues, where we have identified more than 80 different QTLs or genes associated with plant hopper resistance in case of rice. And here, if you can see that most of the genes of the QTLs identified have been clustered in 3 regions. That is cluster A, cluster B, and cluster C. And this makes the possibility that, as I was saying previously, identifying the same region using a different introgression line, or identifying the same region using a different insect species. Say, for example, identifying pH, BPH gene using BPH, and identifying the same region using WBPH or whiteback or BP for copper is also possible. And and with the identification of so many number of genes and introgression of the identified genes into allied cultivars, but still we are like facing the problem of insect resistance and disease resistance. And we cannot like exactly say that this is the reason for the adaptation of an insect or the adaptation of a disease. Like there are so many other factors which are contributing for an insect or an disease strain to get easily adapted to particular resistant gene. And one strategy would be of remedying of resistant genes, which has been commonly done in case of disease resistance, where you permit different genes into a cultivar, and you can see you can see a cumulative or an additive effect where than when compared to the monogenic lines. And that is also true in case of plant hoppers, where like combining of BPH25 and BPH26 had a higher degree of resistance than their monogenic lines. But can we say that it is a durable resistance? Still, we do not know how far this resistance is going to be taken up by the insect. Like because the main thing is like we are poor in understanding the mechanism of interaction. Like if we like if you ask me like do you know the mechanism of BPH26 which is being promulgated along with BPH5, the answer is no, we do not know. We do not know the clear kind of interaction between the insect as well as between the plant that is happening. And now, I mean, how these genomics, I mean, the rice genomics could be used in understanding the plant or insect disease resistance mechanism. I have made a pipeline, maybe it is useful for beginners, where you start with a strong phenotype data that where you screen out your accessions for, say, for example, biotic stress. You screen your accessions along with the control, find out an accession that shows a tremendous change when compared to the control. And you further go for lab bioassays and confirm that the accession is performing well, and do some DNA-seq or RNA-seq analysis, and get the data and map it to a reference sequence, and do some differential expression analysis. Try to identify the differentially expressed genes, and also like after that, try to identify the candidate gene which is involved in the resistance of the tolerance mechanism, and then go for some in silico works like trying to find out how this gene is interacting with your particular with the other genes in the network, and also like develop near isogenic lines where you have the particular candidate gene alone in your near isogenic lines, and try to find out how it interacts with your particular stress factors. What changes do you see? This is the way like how we study molecular interaction. I mean, molecular interaction that gives a more detailed view of what is happening. What is happening when when you introduce a candidate gene to a in a near isogenic line. And yeah, and also this RISE genomics data has provided us so many information on extension markers like SNPs and SSR. And in case of biotic stress, we have this RISE stress resistant SNP database where like more than 400 stress resistant RISE varieties are available. And they list out all the SNPs in their database. And for example, if you want your cultivar is PPT33 and search for PPT33, they give you the total SNP count in that particular cultivar along with the number of chromosomes. And based on that data, you can further proceed with how to correlate your SNP with the stress factor you are working on. And the best example for SNPs in rice biotic resistance is that was identified in rice blast disease, where this particular blast disease was showing 99% homology with the Japanese waxy cultivar, and there were 4 nucleotide differences in this coding region, and 6 were in the intron region. And this particular allele was very rare in nature. That there is an alanine that there is an alanine rather than a serine at position, and that has contributed to the resistance to major pathotypes of fungal blast disease. And this is the best example for rice blast disease. So, like so far we were talking about the biotic stress associated genes. Like, can we say that these genes are like functioning in a very distinct way, or do they do not have any kind of interaction with the the abiotic stress response pathways? The answer is no. As I was previously saying that biotic and abiotic stress response pathway are not a distinct pathway. There are molecular regulators and players which are like helping the plant coming out with a tailored defense response, and we are in the beginning stage of identifying the that tailored defense response in plants. And a very good example for this is that in whiteback plant hopper, which was which was cultivar pre-infested with whiteback plant hopper, was showing much more higher resistance to rice blast disease. And the studies said that the saliva of the whiteback plant hoppers was having some effectors which was injected into the plant, and this effectors had a priming response in the plant, resulting in production of more pathogen responsive genes like pathogenesis like PR proteins and others. And I should also note here that mostly that whiteback plant hopper and I should also say that there are many molecules, and this is a and I can also say that there is this is a pictorial representation showing that that each of the pathways are not like distinct pathways. There are interrelations between them, where like the molecular regulators are regulating each pathway in coming out with a tailored defense response. And I think rice genomics is sequencing of rice genome and available of numerous sessions data is actually helping us to identify like how this interactions happens in multiple stress conditions, and what are the molecular players that are having a role in biotic and abiotic stress factors. And I'm not going in detail into it because we are in the very preliminary stage. And now to summarize my talk, like as I was saying that rice genomics had actually fastened the author-assisted breeding and the breeding. And it also has led in the characterization of core and mini germplasm and total resources for rice genetic enhancement. And genome-wide association studies has been made possible using rice genome data. And the transcriptome analysis has provided a snapshot of all the participating genes, and this can help us in the functional and the regulatory elements that are involved in stress factors. Yeah, and thank you. I think I can take questions now. It's a very basic presentation for the students, like trying to say how a basic research in using rice genomics could be done.
Thank you, Dr. Srivasana. It was a very nice presentation indeed. And you have started from the very beginning by introducing of the introduction of the topic. That was a very highlighted part of the presentation. So, Dr. Arpit, are there some questions to be asked? Engineer Arpit, have you some questions to be asked? Ma'am, I have one question. That in the interaction of insect with the plant, what is the role of gut microbiota?
Interaction of insect with the plant. So, like actually, like I mean, we also have done a study on that. I think I didn't show that. So, when you change a host plant, say for example, you have this green leaf hopper and you are feeding on a particular resistant variety, and when you look for its gut microbe, the gut microbial community is entirely different on a resistant variety when compared to a susceptible variety. The diversity itself changes. it could not be seen in a particular generation. Like when you try to grow the insect on the resistant host plant, say for example 6 to 10 generations. On the 10th generation, the insect is able to adapt to the resistant plant as well as it cuts microbe changes. And I believe that the gut microbe is actually helping the insect to get adapted to the host plant. And we have actually published a paper in Ecology and Evolution where we say that green leaf hopper gut microbiota, the changes when feeding on a resistant host plant. It was published in 2020, sir. Hello? Hello?
Okay. Thank you. Any further
sir,
questions,
we have some
Dr.
questions.
Yeah. Please ask.
Yes, sir, we have some questions. Here, Dr. Manish Kumar is asking, what type of considerations to take into account when comparing different plants of different variety?
No, I'm not getting the question, sir. Can you make it one?
He's asking what type of considerations should take into account when comparing genomes of two different plants of different variety.
Say for example in biotic stress, like comparing the genome would not be a valid thing. I would, as I was saying, that when you have a near isogenic line, that would be far better to compare the genomes, because like you have the entire 99% of the parent except 1% is your gene of interest. That would be a better thing than for comparing genomes of different cultivars in case of the biotic stress tolerances. Okay.
Thank you, ma'am, for providing the answer. And next question we have that how jasmonic acid signaling pathway will playing an important role in abiotic stress in plants.
So actually, that's what I said. Like jasmonic acid pathway is previously reported to be involved in priming responses, like in priming response in plants as well as in chewing and feeding insects. It was the main phytohormone. But like as I was saying, that each of the pathway is not a distinct pathway. That is like jasmonic acid only does this, salicylic acid only does this. That is not the case. There are interaction between the different pathways, and I'm not sure how this interaction happens. I mean, we are in the very preliminary stage of that, sir.
Thank you, ma'am, for your answer. Over to you, Dr. Pradeep, sir.
Okay, if there are no further questions, then I must thank Dr. Srinivasan. Thank you, ma'am, for a very excellent and nice presentation.
Thank you, sir. Thank you, sir.
Then this is the end of the today's session. We had three very eminent scientists for the today's session. That it was started with Dr. Anil Kumar Singh from IIAV Ranchi. Then it was the second speaker of the today's session was Dr. Rajiv Nayan. And the third was Dr. Srinivasana. And before we come to an end, I will give my concluding remarks for this session that global climate change is predicted to impose a severe threat to agriculture productivity worldwide and thereby challenge food security and nutritional security. Technological advances, particularly in genomics or transgene-based strategies, can facilitate the development of some elite genotypes for adverse environment with durable adaptation for the climate change. And genomics in particular is predicted to play a significant role, it is a key point, but we should always realize the role of physiology there everywhere. Significant role in the development of climate change resilient crops. And therefore, the workshop number 3, entitled Genomics-Assisted Strategies for Climate Resilience Crop, is the need of the hour to adopt and integrate genomics technologies along with other modern science tools. And availability of genomics tools and technologies is not a challenge anymore. However, one should be empowered with scientific insight to decide which tool to be used for a particular purpose. And the tool should be integrated in their crop improvement program to accelerate development and delivery of climate resilient crops. Such training courses and workshops, they can help the students, and they can make a collaborations with different partners in the interest of developing climate resilient crops and in addressing the issues of food and nutrition security. So this is my concluding message. And I would like to thank the organizers, especially my very good friend Dr. Kamlesh Chauraji, for giving me this opportunity to chair and convene the session. I am highly very thankful to all the speakers of day 1 and day 2. And not but the least, I will also thank Dr. Srivastava, Dr. Arpit Srivastava, for moderating the session and assisting all the speakers for their troubleshooting problems. So with that, I will end up here, and over to you, Dr. Chauraja now.
Thank you very much, sir, and very good afternoon to Dr. Suja. And I think the session is very, very nice for all of us as attendees. I was also attending the session, and we have learned lots of things from our eminent speakers. And I expect that from all the attendees that they have learned many things to advance their biotechnologies in their research area by learning all this technology from our eminent speakers. So thank you very much, Dr. Pradeep Shukla sir, for accepting my request to be as a convener of this whole workshop on genomic assisted strategies for climate resilient crops. And in future we will again arrange a detailed program related with this technology. This is because this is our need of this present situation. So thank you very much, Arpit and Vivek. They are giving whole time today on behalf of me because I am late in the room to join. And I also thankful to all our speakers, especially Dr. Anil Singh was today here, Dr. Rajiv Bahuguna sir is here, and also Dr. Thanga Suja Srinivasan madam is here, because they have taken all the pain to take session on this Sunday as well. So thank you very much, ma'am, and thank you very much, sir. And we will be in contact with you all in future for our next programs. Hopefully after COVID situation, we will invite you all at our university campus so that we can interact each other. And thank you very much. So Pradeep sir, thanks a lot once again, and thank you all the participants to.
Pleasure is all mine, Dr. Chauraja. Pleasure is all mine.
Yeah. Thank you, sir. So because Pradeep sir is like my big brother, he always support me a lot in all the activities related with many programs and other research activities we conducted here. And thank you once again, Suja madam, Raju sir, and Anil sir. And also our last day speaker, Dr. Jena was there. He has given very good insight in during related with the particular topic, and he has shared all the experience throughout the life to us. And also Rohit sir was there, and Dr. Hibzur Rahman was there from UAE. So overall, I think this whole program is done very
fruitful.
All things were managed by Dr. Pradeep sir very well. So all credit to goes him for arranging the whole program. And thanks a lot. So Arpit,
thank you. Yes, sir.
Now I'm giving mic to you to close this session. And
Sure, sir.
thank you.
Thank you all the attendees and participants. Get ready for our next session, which is going to be held from tomorrow. So all the links and information will be provided to the Telegram link. So kindly be active there. Thank you, everyone. We are now signing off. Thank you, Dr. Pradeep Shukla, sir.
Okay, thank you, Arpit.
Okay, sir. Have a nice day, sir. Have a nice ma'am. Thank you.
Same to you, sir. Same.